Okay, so now let's read this question and let's try to answer it together. So here in red, in this question, I tried to highlight the more important parts of this question. So let's read it together. In addition to the activating or inhibiting enzymes through allosteric regulation. So in addition to allosteric regulation, yeah, what other means, so what other mechanism does a cell use to control enzymatic activity? So again, in addition to allosteric regulation, is there another mechanism that the enzyme use to control the enzyme activity? And here there are many options. And here, remember, they are not asking you what increase or reduce the activity of the enzyme. No, they're asking you how a cell is able to properly control the enzyme activity. Is different, yeah? So thinking about that, yeah, what is the mechanism that the cell reliably is able to control the enzymatic activity? There are many ways in which you can increase or reduce the enzyme activity, but in which way the cell is able to do it consistently. Okay, so this is a clue. So now read it. You can stop the video, and later we can go through the answer. So okay, so if you already stopped the video and you already know the answer, I'm going to continue and show you the answer. So the best answer here is A, is that in addition to the allosteric regulation, the other mechanism to regulate enzymatic activity is the location of enzymes into specific organelles or membranes. Yeah, you will see that the other ones, they or don't work, or the other ones are not a consistent mechanism to modify that the cell is able to control the enzymatic activity. So let's start with the first one. Okay, so here is the best answer. And here I show you a diagram that explains that. So here you can see that the enzymes are localized into a specific organelles or membranes. Yeah, so this property is called compartmentalization. So the cell make compartments, and in that way, you can have enzymes in a specific places. So for example, last time we were talking about peroxisomes, and it reminded me like a recycle plan of the cell. All the thing that is toxic that need to be destroyed go to the peroxisomes, and how they destroyed by the molecular scissors of the cell that are the enzymes. So they have enzymes there. If the peroxisomes release all the enzymes outside, it will destroy the cell. And something like that happened with the vacuoles, yeah, with different organelles like Golgi apparatus, the mitochondria, yeah, the mitochondria. Remember that we talked that in the inside of the membranes of the of the mitochondria, yeah, is the key to understand cellular respiration, and there you have enzymes. So I just told you that one reliable way in which cells regulate enzyme activity beyond the allosteric regulation is to create compartments, so it's the process of compartmentalization. And I show you that enzymes are placed into specific organelles, and I talk about peroxisomes, the mitochondria. Here there are other ones like lysosomes, and also in particular membranes. Yeah, and why this is important? Because it prevents that inappropriate reactions take place. So for example, I show you that if the peroxisome releases all the enzymes, it will cause cell death. It will kill all the cells because it will start breaking all the. It has, for example, the peroxisome have enzymes called lipases, so that they cut the lipids. You have proteases, so it will cut the protein, so it will break all the other proteins and it will break the cells. So this is the best way to regulate enzyme activity for the cell is to create compartments. So A is the best answer, and in the following I'm going to show you why the other ones or don't work or they are not doing that reliably. Okay, so why B is not correct? So let's read B together. B says that the B is the secretion of enzymes out of the cell. So here you can see, for example, a vesicle that is releasing the enzyme. So imagine that we have the peroxisomes that have all these proteases, all lipases, all these proteins that, all these enzymes that break lipids. You have nucleases that break nucleic acid. If you release it outside of the cell, you're going to create a problem outside of the cell. So this is not a good way to regulate enzyme activity. It's just to put the enzymes outside of the cell. In this case, it's not a good way to solve the problem. The second option, or the third option, is C, assembly a large aggregates of the same enzymes. So something that happened in multiple diseases is that when you have clumps of proteins that they are all put together, those ones, when they form clumps, they are not assembled in the correct way. These become non-functional and these can create diseases. So large aggregates of the same proteins, generally, this is something that cells try to avoid because it can damage the protein function and or damage the proteins, and this can become actually a disease. Okay, so B and C, they just don't work. And you will see that the other two options, that is modifying the pH or modifying the temperature, definitely is going to control, to change the enzyme activity. Yeah, but it's not what cells do reliably. Cells don't change the temperature or the pH. Yeah, they don't do that to um to control the enzyme activity. What they do is to put enzymes in compartments, and there they can change or the pH or yeah, in that specific compartment. So, for example, altering the internal pH, generally the cells. So if you see uh, cells generally in the cytoplasm have a stable pH, yeah, and large changes would be harmful. So what they do, if they need to have a lower pH, they have an organelle, so they create a compartment where the pH is different, like the peroxisomes, where you do that. So it's not only at the level of the digestive system, yeah, that you have different pH in the stomach, in the intestines, yeah, in different parts there. Here, this happen at the cellular level, yeah. So this is something that happened at the system, at the level of at a different hierarchical level. It also happened at the cellular level. Another option will be E, that is altering the cell temperature. So this definitely is going to modify the uh, regulate the enzyme activity. However, this is not a safe way or a practical way to regulate the enzyme activity. It can be actually dangerous. And if you increase the temperature too much or reduce it too much, it will definitely or create a denaturalization of the protein. So this is not a way that cells use generally to regulate the enzyme activity. So as you can see, the compared the to A compartments in the cell is the best way to regulate enzyme activity. Okay, I hope that was clear. If you have any question, just let me know.